All right, welcome everybody. It really is my extreme pleasure to welcome Jerry Stein as our speaker today. This is our first meeting in Grand Rounds of the research portion of Grand Rounds, and it couldn't be more appropriate to have him come and visit us. He's from the University of Vermont. He's currently the chair of biochemistry as well as the director of the Vermont Cancer Center. So his plate is full. He came from the University of Massachusetts Medical School, where he served as chair of the cell biology department. I believe he was also director of that cancer center for a few years, and had the Hydak Distinguished Professor of Cancer Biology chair. And he's also the founder of the UMass Human Stem Cell Bank and Registry. So, and to say he's active in his research is clearly an understatement of gargantuan proportion. Just a couple examples. He has 45 basic scientists and physician scientists that he works with in his research. And he has 2 program project grants, several R01 grants. He has over 800 publications. It's absolutely amazing. And his interest has been in multiple areas, in stem cell biology and in skeletal biology, regulation of cell cycle. And he talks about the fact that he was in Dr. Baserga's lab at Temple and was looking at pathology sections and looked at the cells and thought, you know, some of those cells are doing something different. And that essentially stimulated his interest along with other interactions to make him, to start him on his career. One of the things I really like about this is he's going to talk to us a little bit. I asked him, well, talk to us about proteins. And we all know proteins cause cancer, not genes. Just to clarify that for anyone. There you go. So at any rate, he's going to talk to us about the complexities of how genes are regulated, what they produce, and it's just a really treat to have you here. So please.
Thank you.
Let's see. Do we have the take home?
It's okay.
Well, first of all, thank you for the really kind introduction, and thanks for the opportunity to go ahead to visit and spend time with people here. What I wanted to do is to try to give you an overview of approaches that our research group has developed over a number of years to try to understand how you actually compartmentalize regulatory machinery within the nucleus of a cell, and why that is incredibly critical. Is absolutely critical for biological control to proceed during development, which is your remodeling, but equally important, the onset and progression of cancer is a very regulated process. In many cases, what you're really doing is dedifferentiating, and it doesn't happen without a number of control at a number of different levels. And the cell cycle, regardless of the modifications that take place, must occur in a way that is exceptionally competent, perhaps more competent than when you are looking at a normal diploid cell. And that is the basis that's really controlled at the genetic level, but also at the epigenetic level. And why at the genetic and epigenetic level? Because any change that a cancer cell is going to acquire is going to have to be sustained so that the progeny cells are going to be able to retain it and are going to be able to pass it on to their progeny cells. If not, cancer can't be successful. On the other hand, the more we understand about some of these genetic mechanisms and epigenetic mechanisms, that provides insight into a whole series of druggable targets, some of which are being addressed, and others which are probably very viable options. But, and that's a big but, the most important thing is what I'm going to be describing today reflects a long-standing partnership with these people for many, many years. And these are graduate students and fellows in the research group who are not responsible for the studies I'll be describing. More important, they are the people who really designed the approaches that we were taking. So we've been interested not just in genetic and epigenetic control, but how you actually organize regulatory machinery in microenvironments within the cell nucleus, and how you retain those microenvironments during interphase through the cell cycle and then during mitotic division. And that's very critical because you completely dismantle the organization of the nucleus and quickly reassemble it 45 minutes later. And it has to be completely in place and completely competent. And I mentioned a couple of minutes ago that both the onset and progression of cancer are involved in genetic and epigenetic changes, and I'll try to give you examples of how those can be looked at from a regulatory perspective, how we can begin to understand how there is a critical requirement to architecturally organize those, and how, if we play around with those, we can actually modify the properties of a tumor cell. So genetic is obvious. I mean, DNA encoded regulatory information. Epigenetic is non-DNA encoded regulatory information. But the list of epigenetic mechanisms is beginning to grow very significantly. I mean, DNA methylation is the classical one. These are modifications, methyl groups added to DNA. Histone modifications, acetylation, methylation, phosphorylation, poly-ADP, ribosylation. And these are very, very critical. And the reversible processes is very dynamic. You add and you subtract depending upon the physiological cues that the cell is going to receive. And RNA is, for a long time we've understood that RNA regulates RNA-mediated silencing, X chromosome inactivation by exist is something which has been known for a long time. MicroRNAs, now long non-coding RNAs, there's an awful lot that we're beginning to learn about epigenetic control that's RNA-mediated. And over the past few years, one of the observations that our group made was the fact that the transcription regulatory machinery remains associated with genes during mitotic division, actually remains associated with the target gene loci on the mitotic chromosome, and that's a phenomenon that we refer to as bookmarking. And actually the term bookmarking was developed originally by Jerry Workman to refer to histone modifications that are retained during mitosis. And what we did is just added on to that the fact that transcription factors remain associated with the target genes on mitotic chromosomes as well. And that is not just an isolated observation, but that's true for all of the tissue-specific genes that are controlled, that are transcribed by RNA polymerase II, but also by the ribosomal genes that are transcribed by RNA polymerase I. Now, the fact that regulatory machinery is compartmentalized is something which has really been known for a long time, but it's illustrated at a number of different levels. First of all, if you look at information encoded in the DNA sequence, obviously that's very key, but there's structure to the DNA as well. And here it's being drawn as a linear structure, but it gets far more complex than that. You package DNA into nucleosomes, and that in turn into chromatin, and that is not simply a seven-fold compaction of the DNA, but it is specific differences in particular regions that are going to determine accessibility or lack of accessibility to regulatory factors. And then you've got higher order nuclear organization, which are a series of loop domains, and now you have to be able to account for interaction of sequences that could be tremendous distances apart. This is what we refer to simply as intra-chromosomal interactions. They're very specific, very critical, very dynamic and change with the onset and progression of cancer, but it is also becoming evident that there are inter-chromosomal interactions that are very significant as well. And anybody that doesn't believe that there are interchromosomal interactions, then how else could you explain that 17% of your AML leukemias are 821 reciprocal translocations? So obviously during interphase, 821 have got to be in close proximity and at some point have to make contact. And that contact is not through nucleic acids, it's through proteins. Okay, so nuclear organization. I said that there's compartmentalization of regulatory machinery within the nucleus. And what you're looking at here, and I don't know, do I have to kill the lights for people to see the fluorescent images? I can just say, trust me, it gives me more Latin to consider. But what you're looking at here are fluorescent images of proteins that are involved in a whole series of regulatory processes. And the message is very simple. These are not overexpression experiments. You're looking at the endogenous representation of proteins. And what do you really see? AML transcription factors, about 150 to 300 of these foci punctate within a nucleus, compartmentalized. Chromatin remodeling factors, splicing factors, DNA repair factors, factors involved in habitosis, what you can see is that the common denominator is all of the regulatory machinery for all of the processes that are occurring in a cell are going to be punctate, they're going to be compartmentalized. And the complexity of nuclear organization is reflected by the fact that you can superimpose all of these images right on top of each other, because at any point in time, all of these pieces, components of regulatory machinery are going to be operative. So that's the complexity of regulation within a nucleus, and that's essentially the way in which it's actually organized, at least at the descriptive level. I mentioned earlier the fact that the components of organization are dynamic. And I can illustrate that very, very simply for you. What you can see here, this is just through fish. The two red dots are the H4 human histone genes. And what you can see in green is the principal regulatory protein that turns it on at the onset of S phase. And look what you see at the G1 S phase transition. You see yellow, which means that the factors are going to be binding to the promoter, and that domain is present. They co-localize. That's a nuclear body we call a histone locus body, persists throughout S phase, and when the cell enters G2, you can see it dissociates again, and the red is there, the green is there, but they're totally separate. And this is absolutely required for the onset of S phase. Happens about 9 hours after the completion of mitotic division, which is when S phase will begin in a normal diploid cell with a 24-hour cell cycle, or most cancer cells that are continuously dividing and have a 24-hour cell cycle. However, if you take a human embryonic stem cell, that cell has a 12-hour cell cycle, and you almost completely eliminate the G1 period. And now what you can see is that about 2 hours after the completion of mitotic division, these histone locus bodies assemble, the complexes come together. So instead of waiting 8 or 9 hours, it happens right when the cell is going to go ahead and exit mitosis. What that tells us immediately is that there is a clear critical requirement, there's an obligatory requirement between the assembly of these microenvironments and regulatory complexes for fidelity of DNA replication and mitosis. So that gives you sort of an overview of the way in which regulatory machinery is organized within the cell nucleus. And I began by saying that from this type of organization, you have a lot of potential in terms of cancer diagnosis and cancer treatment. And what I've tried to go ahead and summarize in this table is that every level of nuclear organization, from just the DNA sequence to chromatin organization to higher order nuclear organization, already constitutes druggable targets. In fact, each are really targets that have been successfully used for druggable identification, and they're becoming standards of care. Most of your conventional chemotherapy of leukemia and solid tumors is really based on DNA structure and chromatin organization. There are a lot of HDAC inhibitors and DNA methylation inhibitors that are standards of care. And even if you look at higher order nuclear organization, from myelocytic leukemia, what you see is the transition from 2 or 3 PML bodies to anywhere from 930 PML bodies, and you treat the patient with all transuranic acid. Patient goes into remission, and you go right back to the reduced number of PML bodies. Relapse is associated with now a dispersion of the PML bodies and their contents, and so it is very descriptive from a diagnostic standpoint, and from a mechanistic standpoint, it has been able to be utilized for druggable targets. So what I wanted to do is to give you a couple of examples of how we architecturally organize both genetic as well as epigenetic regulatory information. The epigenetic architectural organization is really nicely illustrated by the fact that you actually organize and strategically locate all of the machinery for DNA modifications and histone modifications at strategic sites along the promoters of genes, and that's how you can modify them to be able to accommodate physiological responsiveness. And then you retain the transcription factors at the target gene loci with components of the epigenetic regulatory machinery, so progeny cells receive that information. So let me go ahead and show you a couple of examples of how some of those conclusions were drawn and give you an idea in terms of the directions that some of that research is going. So what I'm going to do is to focus on one family of transcription factors, and these are referred to as RUNCs, AML regulatory proteins. They're involved in osteogenesis, chondrogenesis. They exert positive control and negative control, but they're also involved in myeloid differentiation and play a critical role in terms of leukemiogenesis. So let's take the simple level of organization, and that is the presence of these three transcription factors at multiple sites on the promoters of a target gene and illustrate what that means. This is a simplified representation of what you actually see on the promoter. In this case, it happens to be a gene called osteocalcin that encodes a bone-specific protein, and is also a gene that's expressed in metastatic breast and prostate tumors. But if you look at the promoter of that gene, this is really the proximal region, the on-off switch. This is the upstream enhancer region. You can see there are three sites where that factor binds to. The critical point here is that it is a scaffolding protein. Through a whole series of protein-protein interactions, what you're seeing is a localization of factors that will modify histones, in this case transferases, deacetylases, vitamin D receptor binds there. You can find kinases, you find phosphatases, steroid hormone receptors. And this is really a paradigm for how most promoters are organized. So just looking at the linear structure, you have multiple sites where your tissue-specific transcription factors bind. They, in turn, are going to scaffold a lot of the epigenetic regulatory machinery. That allows you to selectively modify this region as opposed to this region. It means you can determine if the gene is on or off. You can determine if the gene is going to be expressed at a maximal level, if you're going to dampen it down to basal level. And that happens by modifying the chromatin organization and the higher order organization. But it does it by discriminating between which of the epigenetic enzymology is going to be bound at each of those sites. So that illustrates how, if you look at the promoter, what you can see is a blueprint for the ability to respond in a regulatory manner. Let's look at the level of higher order nuclear organization, at the level of observing the compartmentalization. If you simply you treat with an antibody to AML, this is not an overexpression experiment. This is actually a direct photograph. It's not even a captured image. And if you look at AML, what you can see is about 150 to 300 of these foci within the nucleus of a cell. Now, that tells us something immediately. First of all, if you could visualize it, it means you don't have a single molecule. You've got an awful lot of these molecules that are present in a limited number of sites. But the thing that bothered us at the onset is we always found a range of 150 to 300 of these foci. And you would like to understand, what does it mean if you have 150, what does it mean if you have 300? Because we're looking here at normal diploid cells. And we saw this with myeloid cells with the AML1 factor, and we saw it with bone cells as well with the other RUNX transcription factor. So that was a puzzling problem, why you had this variation. The second thing was our curiosity was tweaked in terms of how do you actually organize those foci? Because we know that a transcription factor binds to a site on the promoter utilizing a DNA binding domain. We know that a transcription factor, that protein enters the nucleus utilizing a nuclear localization signal. But how do you get from the point of nuclear entry to the promoter site on the gene? So we went ahead and took the approach that a molecular biologist would take. We already knew that the functional map of that protein was that the N-terminus had the DNA binding domain and a nuclear import signal. So we started simply making deletions to the C-terminus and asked when we lost the capability to be able to localize within a nucleus. And we found that when you eliminated that RNA sequence, all the protein was in a nucleus, but none of it was punctately organized. And if we took that 31 amino acid sequence that we called the nucleomatrix targeting signal and put it onto GAL4, here's the same punctate pattern you see for the wild type protein It completely co-localizes. And that indicates that there is a region of the C-terminus that we were able to refine down to 31 amino acids that was capable of directing that factor to a particular site within a nucleus. And when you mutated that sequence, or when you deleted that sequence, you observed about a 10-fold decline in transcription. So we then went from fixed preparations to a live cell imaging approach, where we put a fluorescent tag, green fluorescent tag, on the AML protein, looked at it over 30 minutes by time-lapse cinematography, and you can see that the foci are there. There's your 150 to 300 foci. You could actually compare them from spot to spot. So they were reasonably stable. If you mutated that subnuclear trafficking signal, all the protein is there, smeared all over the nucleus. Some of it is out in the cytoplasm. So that told us that it is in fact involved with localization. And so the question which then arises is there specificity, and can you actually quantitate the phenomenon you're looking at? So in collaboration with an X-ray crystallographer, the crystal structure is resolved. It's got a V-shaped structure with a flexible glycine hinge, and you have two interaction domains, which single introduction of single amino acid substitutions there is going to almost completely compromise fidelity of nuclear localization. Instead of co-localization, you can begin to see now red and green are showing up, but you get even a more pronounced change in the effect on transcription. So those pieces of information together told us that if you are interested in understanding mechanistically how you organize regulatory proteins within a nucleus, you have minimally three signals that you have to deal with. You've got a nuclear import signal, you've got a subnuclear trafficking signal, and then a signal to interact with that sequence on the promoter site of the target gene. So that gives us just the beginnings of understanding mechanisms that account for some of those diagrams that I showed earlier. But what I described to you so far with respect to nuclear organization and the trafficking mechanism occurs within a cell. What happens if you're looking at an intact organism? What would happen if you were to perturb capability to correctly traffic? What will that do to development? So there was a really nice opportunity to be able to address that question. And that's because having shown that RUNX AML controls skeletal genesis, and actually very soon after we identified RUNX AML as the bone-specific transcription factor, two groups did a knockout. The knockout phenotype was really, really profound. Here's your normal skeleton, cartilage is blue, and bone is red. And if you knock it out, you get a very nice cartilaginous skeleton with very, very minimal bone formation. So we went ahead and did a gene replacement, a knock-in, where we substituted the wild-type gene with one that has a mutated subnuclear trafficking signal, and you get exactly the same phenotype as you do with the knockout. And there's actually a heterozygote phenotype where you lose the clavicle and you get that with and you get exactly the heterozygote phenotype as well. So that told us that the presence of the factor is not sufficient to be able to support biological control even during development, but there's a requirement for it to be located at the right place. And let's look at myeloid differentiation that's also controlled by the RUNX AML transcription factor. And if you put a single amino acid substitution in that subnuclear trafficking signal, you no longer go from your blast cell to your very differentiated myeloid cell, but you have a phenotype which is very similar to a blast cell and very analogous to a pre-leukemic state. So these are examples that give us some confidence that there's an actual trafficking mechanism that is obligatory for both biology and pathology in an intact organism or within an organism when you're looking at myeloid differentiation. But if that type of exquisite nuclear organization is so darn critical, what happens to it when the cell goes through mitotic division? because you have a requirement apparently for it before mitotic division, you have a requirement for it immediately after mitotic division, what happens during mitosis. So there are really two models that you have to distinguish between. To degrade, resynthesize, assemble, and organize all of that regulatory machinery the minute the cell emerges from mitosis, or do you retain it and simply partition it to the progeny cells? So the experimental strategy is as straightforward as it gets. Instead of looking at cells that are exponentially growing, you just look hour by hour through the cell cycle and look at your fluorescent images. And right before mitotic division, if you look at a G2 cell, and you look at the myeloid or the osteogenic AML factor, you no longer have that range of 150 to 300, but what you find is you have 300, and here's the progeny cells that each have 150 of these punctate foci. And if you count it, this is essentially what you see pre and post-mitotic division. So that told us that you apparently, but it's not formal proof, partition it to the progeny cells. But what happens during mitotic division? And the approach, I mean, is even simpler than this. Just look at cells that are dividing and see what you can find under the microscope. And this is essentially what you find. This is the cell in G2. This is the cell undergoing mitotic division. Blue are the chromosomes. You can see the foci on the chromosomes. You could see the others on the microtubules. If you count the 300, you could find the 300 are sitting here. Through biochemical analysis, you can show this is through a microtubule binding domain. This is going to be through the DNA binding domain. And if you look at this really closely, you'll see that each of the foci of the chromosomes are duplexes. They're not single foci. I'll show you some images that I think illustrate that even more convincingly. But as the cell goes right through mitosis from prophase to metaphase to anaphase to telophase, you can see that the foci are going to be retained. If you mutate the DNA binding domain, it's no longer associated with the chromosome. And if you do metaphase chromosome spread, here you can see that these foci are symmetrically located right on the sister chromatids. So that was the data that suggested to us that you have mitotic bookmarking of transcription factors and that the factors are going to be retained with their target genes during mitotic division. But an image is suggestive that it's a sequence-specific interaction. What we really wanted to be able to do is to establish that during mitosis, these foci represent the factor associated with their actual target genes. So we used a genomic approach to be able to document that. And the approach was very simple. We asked what is expressed prior to mitosis, right after mitosis, that will not be expressed if you do an inhibition, a knockdown of the AML factor. And we identified 40 genes that fit those criteria. This is from an unbiased screen. And then when we looked at those 40 genes, 38 of those had multiple AML sites on their promoters. And we did the chromatin immunoprecipitation analysis of mitotic cells. We were able to confirm that indeed all of them had AML associated with the target genes. So that was the basis for the conclusion that transcription factors are going to remain with the target genes during mitotic division so that post-mitotically the regulatory machinery is in place. What surprised us, and this was not clever insight, this was an observation that was really perplexing at first, but what we found was that both the myeloid and the osteogenic AML factor was associated with the ribosomal genes. And we found it associated with the nuclear organizing region. And this is a gene that controls Pol2-driven transcription, and ribosomal genes are transcribed by RNA polymerase I. So that was the first inclination that actually there may be factors that control both Pol1 and Pol2 genes. So we went ahead and looked at all of the acrocentric chromosomes. Remember, there are 5 acrocentric chromosomes that have the ribosomal genes associated with them. And by the way, each of those nuclear organizers have 100 ribosomal genes in tandem. And you can see, indeed, the foci are symmetrically located right on the sister chromatids. And what you see co-localized with it is UBF1. That's the Pol1 co-regulatory factor. And when the cell exits mitosis there, you can see AML and UBF right in the periphery of the nucleolus, which is the region where ribosomal genes are actively transcribed. And if you do immunochemical labeling, you can show that it's present indeed within the nucleolus. So we went to a molecular approach, and we did chromatin immunoprecipitation analysis and showed that there is in fact occupancy of those sites, those AML binding sites on the ribosomal gene promoter. We did what they call a rechip, simply to be able to show that you actually have a complex of AML and UBF, and indeed all of these sites are going to show occupancy of both components of the regulatory complex. And if you do either inhibition experiments or you do upregulation experiments or expression experiments, you can modulate ribosomal gene transcription. Here what you're doing is either dampening or upregulating RUNX expression. And there's a very striking effect on the level of protein synthesis. So it's not just controlling expression of the ribosomal genes, but it's also having a very direct effect on capacity for cellular protein synthesis. So this was information that you could easily take to begin to think that if you take a tissue-specific transcription factor like RUNX AML, you're regulating RNA polymerase II genes, you're regulating cell cycle mechanisms, a series of those, you're controlling phenotypic genes, but in addition, you're also modulating cell growth by impacting on the control of protein synthesis. So the obvious question was, is this a mechanism which is confined to the RUNX AML family of transcription factors, or is there really broad-based applicability to that type of control? And the answer is it seems to be quite broadly applicable. We're able to show that that's true for the RUNX AML myeloid genes, for the skeletal genes. It's also true for another component of the AML family, RUNX3, which is involved in gastrointestinal and neurological control. But if you look at leukemia, if you look at AML leukemia, where you have an 821 translocation, retaining the DNA binding domain, picking up a piece of chromosome 8, losing the subnuclear trafficking signal, but acquiring a subnuclear trafficking signal from the chromosome 8 donated piece of DNA. You have a different subnuclear trafficking signal, goes to a different place, and you still are going to bookmark the target genes that are involved in myeloid differentiation, but in this case suppressing rather than activating. So the obvious open-ended question now is, we've talked about a transcription factor that binds to target genes. We've talked about a subset of the co-regulatory proteins. Are all the co-regulatory proteins retained? Are only some of them retained? And the answer lies somewhere in between. And that is, you carry a subset of the co-regulatory proteins, and there's a certain amount of plasticity where you can indeed exchange depending upon what the post-mitotic requirements are. And if they are the same as prior to cell division, then it seems that you reacquire the components of the regulatory complexes that were lost. And otherwise, our adjustments are made. So with respect to osteoblast differentiation, myeloid differentiation, I've already illustrated what you see. If you look at mesenchymal cells going into the muscle phenotype or into the adipocyte phenotype, exactly the same mechanism is going to be utilized. These tissue-specific transcription factors, MyoD or CEP, are going to bind to multiple sites on the promoter of the POL2 target genes that will affect growth and differentiation. But MyoD and CEP are also going to coordinately regulate ribosomal gene expression and protein synthesis. So when you look at the ribosomal genes, you have your tissue-specific factors bound either RUNX, MyoD, or CEP, and then the appropriate co-regulatory proteins. So there is a way to be able to relate control growth and control phenotype. Now, I mentioned the fact that bookmarking is a mechanism for being able to convey regulatory information from parental to progeny cell. But it changes very strikingly when you're looking at a change in the biological situation. That is incredibly incredibly clearly illustrated when you look at early stages of embryonic development. And if you look at a pluripotent human embryonic stem cell, which is a cell that you have unrestricted proliferation with very stringent cell cycle control, and you look at the types of post-translational histone modification that take place, you have certain things which are incredibly unique. What you begin to see is that your bookmarking of single genes is going to occur in a very specific way. You have this phenomenon that's referred to as bivalency, where a number of these genes are going to have both positive as well as negative histone modifications on them. And what you can see here is that the negative modifications are going to be present constitutively, whereas you go and find a subset of these which are actually involved in activation, that's K2 trimethylglycine, is going to be present only during mitosis. And as soon as the cell becomes lineage committed, despite the fact that proliferation continues, it now goes ahead and becomes constitutive as well. So what that illustrates is that when you look at epigenetic control, DNA methylation, histone modifications, and transcription factor occupancy, it occurs in a very physiologically responsive manner. So let's look at the problem of cancer and how, in fact, one can take the epigenetic mechanisms and the requirement for specificity of subnuclear localization to begin to understand some of the pathology of cancer. And we went ahead and took a very simplistic example, and that's based on the observation that for many, many years it's been known that breast and prostate tumors that are advanced and metastasized to bone express a bunch of bone proteins. Osteopontin, osteonectin, osteocalcin. These are biomarkers for metastatic disease. So we asked the obvious question, are these bone proteins in the tumor cells controlled by the same regulatory factor that controls it in bone? And the answer turns out to be yes. AML-RUNX controls the bone proteins in the breast and metastatic breast and prostate tumor cell. So we did a very simple experiment. We went ahead and we asked if, in fact, we were to knock down AML. This is in a orthotopic model of breast cancer. What you can see is that you really hold back the growth of the tumor, and if you upregulate it, you can exacerbate the growth of the tumor. We expected that result, but what we wanted to ask was, can you use fidelity of subnuclear trafficking as a druggable target? If you block the ability of that factor to go to the correct place, could you block the ability of that factor to exacerbate the growth of the tumor? And we did the experiment in a very direct way. We went ahead and we took breast cancer cells, in this case we used MD8-31 breast cancer cells, and you introduce them directly into the tibia. You do an intertibial injection. And you can see that in about 19 days that the tibia is just about completely eroded. And this is very analogous to what you see with breast metastatic disease. If you either put 2-point mutations in that subnuclear trafficking signal, or you eliminate completely, you can see that when you put mutations in 16 out of 20 animals, you have a reasonably intact tibia. And if you eliminate the whole C-terminal sequence, you can see that it is virtually intact in all of the animals. So that tells us that that signal is, in fact, has tremendous potential in terms of being a druggable target. So let's look at myeloid leukemia, and you find a very similar situation. This is just a schematic of the AML factor again. Here's your DNA binding domain, your nuclear import signal. Here's your C-terminal subnuclear trafficking signal, and the 821 translocation, you relinquish this region, you retain this region. Here's your fusion protein, and what you can see is it no longer has the information to go to the correct place, it goes to a different place, and you end up with the retention of blast cells. So the question we wanted to ask was, is that change that you're observing related to the loss of the ability to localize correctly? So instead of looking at the fusion protein, we simply introduced a single point mutation in that subnuclear trafficking signal, a mutation that we had defined from the structural analysis of the protein. And the net result is that it's expressed very nicely. It's now in the soluble nucleic compartment. It is no longer architecturally associated. And the consequences are as follows. You now are going to block myeloid differentiation. This is just expression profiling. You can see that you acquire increased capacity to express genes for the cell cycle because the cell is proliferating, and you lose expression of genes that establish competency for apoptosis. And now what you can see when you actually look at what is happening with the cell, instead of proliferating, ceasing to proliferate, differentiating, and then undergoing apoptosis, the cell continues to proliferate, remains blast-like, and reflecting back on nuclear organization, in other words, instead of remodeling your nucleus like this for a specialized function, you retain this type of organization, and this cell continues to proliferate. So we then asked the question, what about another dimension of control, microRNA-mediated regulation, which clearly is involved in development and differentiation and is very much involved with oncogenesis. And we asked, is there a requirement for fidelity of transcription factor localization to support microRNA-mediated regulation? I'll give you a quick example of what you see there. And so what we did was a very straightforward type of a screen for microRNAs that was based on compromised nuclear organization. We asked the question, what is expressed in a cell that has a mutation in the subnuclear trafficking signal that is also expressed in a cell that has an A21 chromosomal translocation? And these are not cell models, but these are patient samples, patients who have AML leukemia with an A21 translocation that is not going to be expressed in the wild type cell. And when you do global expression profiling, you can identify several microRNAs that fit exactly those criteria. And one particular microRNA, MIR24, was able to go ahead and exhibit expression in the AML patient in the cell that has an introduced subnuclear trafficking signal defect. And when we went ahead and identified target genes for MIR24, the principal target turned out to be a MAP kinase phosphatase. And that became very important because what it means is when you when that microRNA is expressed, you're blocking the phosphatase. The kinase is going, the MAP kinase is going to remain active, and proliferation is going to continue. And if you look at the consequences, what you can see here is that the cell is going to now become cytokine independent and to continue to proliferate and exhibit a significant increase in capacity for uncontrolled proliferation and colony formation. So when you're looking at a regulatory microRNA that is controlled by AML, what is happening is the wild type rungs AML to suppress that microRNA, and that's going to go ahead and block proliferation, and myeloid differentiation persists. You block the phosphatase because now the promoter with the fusion protein bound is going to go ahead and to be active, and you have continued proliferation. The last two slides I want to show you go back to the original diagram and support the statement that every level of nuclear organization can in fact be informative in terms of understanding the pathology of cancer. So what I wanted to do is to show you an approach where you can actually look at higher order nuclear organization, where you can directly assay intra and inter chromosomal interactions and look at that in a normal cell and look at it during a model of breast cancer progression. And the approach is really straightforward. There was a technique that was developed about 3 or 4 years ago called chromosome conformation capture. And what you simply do is you take the entire cell, the entire genome, and it's cleaved with restriction enzymes after you've cross-linked. So now what you've done is you have proteins that are going to go ahead and to be cross-linking. And you, under very, very dilute conditions, you now you now go ahead and permit ligation to take place. So you generate a bunch of circular fragments of DNA that are going to represent the entire genome, but what you're going to be, what you'll have within these fragments are the pieces that were actually linked together by proteins. So these are sequences that can be significant distances apart or between chromosomes as well. You then do genome-wide sequencing analysis and determine exactly what the sequences are. And when you do that type of analysis, and here what we're comparing are MCF7 breast cancer cells and MCF10A cells. So the comparison is a cell which is close to a normal mammary epithelial cell and a cell which is representative of an early breast cancer cell. And the point here is that what you're looking here is the interactions between chromosomes, and if you just expand that region here, in both of these, you can see that there are, in fact, obvious differences. And that's illustrated right here where you're doing a direct comparison. So it tells us that you not only have, during tumor progression, differences in genes that are expressed and proteins that are bound to promoters that are controlling expression, and differences in histone modifications, but also differences not just in locations of genes within the cell nucleus, but actual physical interactions. So it becomes important to understand what's happening in terms of controlling a gene, but it's also important in terms of understanding the environment that a gene is located in. And all of this is extremely important and very, very informative, because each component of organization and control is in fact giving us information about biology and pathology. There are a number of proofs of principle that have been established that give us confidence that this is in fact valuable information for understanding cancer, and a number of standards of care are emerging that are utilizing each of these different levels. So to us, it's obvious that we've been able to uncover a lot more in terms of questions than we've answered, but I think there's also a lot of promise that it's very important to understand how you organize regulatory machinery in the nucleus if you're a biologist or a pathologist. So thank you very much for the opportunity to share this with you. And obviously, I'd be happy to answer questions. Yeah.
So in your intratibial model, you knocked out RUNX2 function and also incorporated some mutations and indicated that that supported the idea that RUNX was a druggable target in breast cancer. Do you have any ideas specifically of how you could drug RUNX2 function? Because of course transcription factors represent the last frontier of druggable targets in cancer. I'd like to hear your ideas of how to interfere with the function.
Well, it's very easy to go ahead to identify small molecules that will interact with it. But the questions obviously then become, number one, delivery systems. Two, how can you get it to the nucleus so that it's going to be stable? And equally important, the problem that you're up against is if in fact you block, are you going to block bone remodeling? I mean, so those are really the issues. So there actually are some examples of small molecules that have been identified and that are being looked at. And I haven't really seen any data that I'm comfortable with that indicates you won't have. You're not going to have off-target effects, but you're going to induce another set of complications.
And so how do those small molecules function? What specifically do they bind to?
I can't really tell you because the small molecule that was identified was not worked on in our laboratory. So all I really have is the statement that it's been done. Khalil?
Cancers that progress in a way, for example, in the case of prostate cancer, it progresses from hormonally responsive to non-responsive. Would you predict that there is a different type of intranuclear organization as it develops, or is it just simply a case of some specific genes being affected?
Well, clearly there's a change in nuclear organization that's been observed. Whether that is functionally linked to the actual, progression of the disease, I don't think is totally clear right now. And then you've got all your genomic and non-genomic effects with the receptors as well, which become important. So there are nuclear, you have multiple nucleoli. And is that functionally linked or not? We're just beginning to go ahead and look at it, but clearly you could actually see a change even in the organization, in other words, of the protein regulatory machinery. Yeah.
So in the context of tumor heterogeneity, where you have populations that survive, are they also experiencing different types of regulatory mechanisms or localizations? Has that been looked at?
It's a really good question. I don't really know the answer to it. There's really a lot of emerging capability in single cell analysis that I think is going to be very informative, where you could look at the total complement of proteins and you could look at the full complement of genes that are expressed. But I don't really think that there's a definitive answer to that at all.
Okay. Are there other questions? Yes.
Yeah.
Does what's a change in organizational numbers during differentiation?
Does it change in terms of
The numbers or distribution in the nucleus?
the numbers of foci?
Yeah.
Good question. The number of foci seems to be reasonably constant, at least in the situations that we've looked at. And if you look at the histone locus bodies that we showed earlier, they remain absolutely constant. Now, the reason that becomes a very important question is that when you look at a cell which is pluripotent, control is totally different than when the cell initiates lineage commitment. Because in the pluripotent cell, you've got a completely euchromatic nucleus with very, very, very little heterochromatin. And most genes are transcribed, they're leaky as heck, and most of the control is degrading the transcript, so that the protein is never made. As soon as a cell becomes lineage committed, what you see morphologically is you start heterochromatinizing, and at that point, transcriptional control begins to start dominating. So it's very different. But when you look at a structure like a histone locus body, you look at a gene like that, you find that it's the same prior to and following lineage commitment. The argument I raise is that cells are always differentiated, but a pluripotent cell is specialized to proliferate, and that is its tissue-specific, gene at that point. You could reinforce that kind of speculation because you could also take a lineage-committed cell and reinduce pluripotency, and then you end up with your abbreviated cell cycle. But you always have, regardless of whether it's a long cell cycle, a short cell cycle, you always have, and if it's heterochromatinized or totally euchromatin, the genes that are expressed will go ahead and undergo remodeling like that, and they're organized the way that's been shown. So the factors are there, and they get reorganized under different circumstances. Ruxolitinib timidopyr seems to have about the same number, but I think there's a lot more to be done to say that with a heck of a lot of confidence.
Is there the connection between proliferation and the different lineages that occur from that? It seems like there would need to be additional tissue-specific factor which is involved in another layer of organization.
Well, the answer is you're absolutely right. There has to be. In fact, that's when these other factors start start coming in, and that's been looked at to some extent. The onset of myogenesis, when myogene comes in and starts assuming the types of roles, it ends up very similar to what you saw for AML. The moment the tissue-specific factor appears, it is controlling both growth and phenotype, at least for AML, for myogene, for CEBP, and it is for the translation, for the fusion protein as well. So it looks like what controls phenotype controls growth.
Are there other questions? Thank you so much.